The ME, CFS and Lyme Association of Western Australia would like to thank Professor Peter Irwin and Dr. Charlotte Oskam from Murdoch University for being here today to present Unravelling Tick-borne Illness in Australia. Professor Peter Irwin and Dr. Charlotte Oskam are from the Tick Laboratory, which is a part of the Vector and Waterborne Pathogen Research Group at the College of Veterinary Medicine at Murdoch University in Western Australia. For the past 4 years, their Tick lab has been studying bacteria and other microorganisms within Australian ticks and have found some potential pathogens. They have recently commenced a new pilot study into the causes of illness in people who have become sick after being bitten by a tick in Australia and are interested in recruiting patients to participate in their research. In today's talk, you'll hear about their research, how they do it, and what they have found so far. So, without further ado, can I please introduce Professor Peter Irwin and Dr. Charlotte Oskam.
Good afternoon, everybody. Welcome to this event. Thank you all very much for coming. I know that for some you've come a long way, and for others it's been just difficult getting here. So I greatly appreciate you attending. And it's wonderful for us to be able to talk a little bit about our research. My name is Peter Irwin, as Stephen said. Thank you for the invitation, for the introduction, Stephen. And we have Charlotte Oskam with us as well. And we work at Murdoch University. So I thought I'd start by introducing our research team. We're based at Murdoch University. So there's myself and Charlotte. We work with Professor Una Ryan. And Una and I have worked together for the best part of 20 years. She is a biochemist and an expert in molecular infections. I'm a veterinarian, actually, by training, but I've also spent most of my professional life in the research sector working with ticks and tick-borne disease, mainly with animals, with companion animals or with wildlife. But more recently, of course, we've become very interested in what could be transmitted to people. And here we've got a list of some of our students. We have a number of PhD students. We have honours students that work with us as well. I should also take an opportunity to thank and to acknowledge the people who have been funding our research. It's very difficult to get research in this area, as you probably know. But we've been very fortunate in the last few years to have a couple of grants, major grants, from the Australian Research Council, together with Bayer Animal Health and Bayer Australia. So you probably know Bayer is a major pharmaceutical company. They work all around the world. They've been working with companion animals. I've known them for a long time because in my work as a veterinarian, I've worked with them researching infections in dogs and cats. So they've been instrumental in funding some of our work. And in recent times too, we've received donations from the Lyme Disease Association of Australia, from the Country Women's Association, and increasingly we're receiving philanthropic and private donations to our research. And we're extraordinarily grateful for that, because the research that we do is expensive to conduct, and without the money we can't do the work. As Stephen said, we're not medical people ourselves. I'm a vet, and I better not give you any advice about your health, but we will talk, of course, a little bit later about some of the problems that have been associated with the testing that you're all very familiar with, I'm sure. But our focus today is going to be predominantly on ticks and our research and what we are finding inside those ticks. So this, if you like, is an overview of the talk we're going to give. I'll say a few words about zoonotic tick-borne illness. So zoonotic means infections that can get into people. We'll talk a little bit about ticks, because that's been an interest of ours for a long time. And particularly we'll talk about the organisms, the microorganisms that can be found inside ticks. Charlotte's going to give you a crash course in the DNA work that we do, the laboratory testing methods that we use, and we'll tell you about our results in the Australian ticks. At the very end, we'll talk a little bit about where this research should be going. Okay, so start off with zoonotic tick-borne infections. Well, this slide, as you can see, has a list of names of infections that in other parts of the world, for the most part, are very well known. You'll be familiar with some of these: babesiosis, anaplasmosis, ehrlichiosis, Lyme, Borrelia, of course, typhus. All sorts of diseases can be transmitted by ticks. In addition to infectious disease, ticks are responsible for causing neurological disease, skin disease, and quite recently this new condition of the mammalian meat allergy has been recognized in Australia and in other parts of the world. So this is an anaphylactic, allergic type reaction to the bite of certain ticks. Not all ticks, but certain ticks. For the most part, and this is one of the issues or one of the problems, for the most part all this research has been done in the Northern Hemisphere. And for the most part, many of these infections are transmitted by a group of ticks, a family of ticks, if you like, and it's called the Ixodes ricinus group. And there is a number of different species of tick, and they occur throughout the Northern Hemisphere, some of them in the United States, some in Europe, and some in Middle and Northern Asia. And it's important to note that whilst these ticks are well known to transmit some of these diseases, and the reason for that is because people have been researching this for 20 or 30 years, really working out which tick transmits which disease and when and how. Much less has been done in the Southern Hemisphere, as you're all aware. And importantly, I guess, none of these ticks occur here. And one of the things we've been looking into is whether in fact some of these ticks could be occurring here. But they don't. No one's found any of these ticks that transmit those diseases in Australia. So in Australia, the situation, as is generally recognized, is that we have a number of rickettsial diseases, so spotted fever and other type of rickettsial disease, Queensland tick typhus, and Coxiella or Q fever is also a tick-borne disease, although humans usually get Coxiella or Q fever from exposure to other animals, so to generally to ungulates, to sheep and cattle during the birthing process, and or in abattoirs. So abattoir workers are at risk of Q fever. Vets and farming people generally are at risk of Q fever. So this is what is recognized at the moment. And of course, I'll talk more in a moment about what else could be here. I thought I'd talk a little bit about ticks. You probably just hate ticks, so this probably doesn't interest you quite as much as it interests me. But I thought perhaps to clarify some of the issues, so we all know what we're talking about, I'd talk a little bit about the ticks. So there are 4 life cycle stages, if you like. Female ticks lay eggs. The eggs develop into the larvae develop into nymphs, and the nymphs develop into either female or male ticks. And up the top here, these are all unfed ticks. But when they feed, as you know, they fill up with blood. Quite often we hear people thinking that these sorts of ticks are different to those sorts of ticks because they look different. But all it is that these, particularly the females, can fill up with blood. So that's the same tick as that, but after it's had a meal. Okay, so they do look completely different. This is obviously all magnified. The larval ticks are very, very small, 1 millimeter in size, generally. Nymphs, a little bit bigger. When they fill up with blood like this, they're probably 2 or 3 or 4 millimeters long. They're getting quite big. And then, of course, the adults probably start off at 8 or 9 millimeters. Sometimes a kangaroo tick, you might have seen, kangaroo ticks that are full of blood, can be almost 2 centimeters in diameter, really quite big and full of blood. So the important thing about these is that they hop on and they hop off their hosts, which are native animals. Humans have no ticks of their own. We don't have the human tick. All the ticks that get onto us by mistake. They've evolved for millions of years to live off native animals. And when you go to the textbooks, all the pictures you see of life cycles are of squirrels and rabbits, northern hemisphere animals. So what I've done here is I've just put in a few of our creatures, because the Aussie ticks will be adapted and are adapted to Aussie animals, of course. So ticks have to drink blood. That's how they get enough energy to live and to molt into the next phase, and to have babies or lay the eggs and all that sort of thing. And they generally have a different host for each stage. So a larval tick can hop onto one host, feed, it falls off, it molts, it'll hop back onto another host, feed, drop off, hop onto the third host. And that life cycle for any given tick can take several years. It takes quite a long time, and it's seasonal because ticks are most active in the warmer weather. Although it's very well known which hosts, so when I say hosts, which mammals, mice or rats or hedgehogs or squirrels in the northern hemisphere, that's really well known. What is less well known is exactly which of our native animals here in Australia host the different tick species. And although all the tick species, more or less, I think, have been identified in Australia now, quite how they live their lives is not very clear. So that's a female tick laying eggs. This is the paralysis tick, Ixodes holocyclus, and they lay hundreds to thousands of eggs from this big engorged body. So when they feed, they fill up with blood. The blood essentially is converted into eggs, and then they lay the eggs, and they eject them out of a little hole underneath here. The ticks then hatch, the larvae, or the nymphs and the adults need to find a host. And this is the dangerous bit for us, because the ticks will go and sit on a blade of grass and waiting for some nice meal to come past, and the meal is the host. And normally it would be a kangaroo or a wallaby or a bettong or some Aussie animal here in Australia, but of course it can be us, and it can be our companion animals as well, and they hop on and feed. We identify ticks with different features. They've got lots of bits and pieces that help us identify what they are. That's an electron microscopy picture, a scan, very, very enlarged of a tick. It's not in very good condition. It's broken a few legs here. But what I wanted to show you here is that ticks in their insides, and this is important from what I'm going to say later, in their insides they have features that are similar to us. They have a gut, of course. They have to, they drink blood, and the blood has to be processed in their system. So they have a gut. It's called the midgut, and then they also end up pooing out the other end. So they have a place that does that. The females have ovaries, and they have salivary glands. And that's important. These salivary glands are important because that's where, of course, organisms are stuck into the host. They're transmitted out of the salivary glands. Unlike us, where our salivary glands are all up in our head, up here sort of thing, ticks have these huge salivary glands that extend down their bodies. This is the kangaroo tick, which would be the most common tick in Western Australia that bites people. This is a female that you can see her running across a piece of material. And look at these very long mouth parts. That's what they sink into people. And they're normal animals. So they have powerful mouth parts. They're horrible if you look at them. They've got these serrated edges, and it's this that they stick into people to be able to feed. And this is magnified again, and you can see this is the part that is stuck in, and these then go out to each side, just like that. And this is a picture taken by somebody in Western Australia. Here's a female kangaroo tick attached with its palps out to the side, and it's feeding. Okay, so that's the life cycle. They feed prolifically, they drink blood. While they're drinking blood, they can transmit infections. So I said a little while ago that the Aussie ticks are unique. Indeed, we've got about, and it does change occasionally as people do find new ones, but we've got about 65 different types of tick in Australia. And as I said earlier, the really important thing is they only occur in Australia. They don't occur anywhere else, like for the most part other people's ticks don't occur here. And that's important from an evolutionary and biological perspective as to what we could be getting from these ticks. And I'll come back to that in a moment. So these ticks have been here a very, very long time, millions of years, and they've evolved with our unique wildlife. And I expect some of the animals that have become extinct, we used to have all sorts of megafauna, a whole load of marsupial animals before now that have gone extinct. They will have had their own ticks, and the ticks will have gone extinct with them. So the ticks that are here, that are Australian ticks, are known to contain a number of different organisms. So we're talking about three broad groups. We're talking about hemoparasites. So hemoparasite, the word means parasite in the blood. And the ones that are really well known include the paraplasms and trypanosomes. So I expect you know that you've heard of trypanosomiasis, sleeping sickness in people. Well, what you might not know is that many of our native animals here in Australia have trypanosomes in their blood. Again, I'll show you a picture of one in a moment. But they're transmitted by ticks into bettongs or into kangaroos or whatever the host is. They've not yet been found in people. There's no suggestion at the moment that they're zoonotic, that they can get into people. But the point I want to make is that the Aussie ticks are known to transmit lots of parasites into native animals. What's less well known, simply because they're harder to see and harder to find, are the bacteria. What is known is that we have rickettsial. As I said earlier, we have rickettsial infections here, and that's fairly well understood now. The viruses are not well understood. Overseas, there's something called tick-borne encephalitis. That's viral disease transmitted by ticks, and to date in Australia, that's not been found. It could well be here. We now know that Aussie ticks have lots of viruses inside them. So again, that's something that I think will become clearer. But the question really is, well, okay, we know about these things. What are we missing, or what don't we know that could be transmitted to people? I just will show you these pictures just to show you how extraordinary some of these things are. These here are trypanosomes. So like the sleeping sickness parasite that causes so much illness in Africa, in people and in animals, many of our marsupials have these things in their blood.
Those are red blood cells.
So these are the red blood cells here, okay, blood corpuscles if you like, or red blood cells. These funny looking purple things are the parasites. Okay, these are white blood cells. This is actually from a platypus. Okay, and the platypus has its own tick. And the platypus has its own trypanosome. And I guess, again, it's an example of how these things have evolved together for an awful long time, because these do not seem to make platypuses sick. That's the other thing, that the wildlife have these organisms. There's another one. So these are red blood cells. There's a parasite living, that's like a babesia. And many of you know what babesia is. It's a protozoan parasite that lives in blood cells. Well, there is one that lives in this marsupial. This is a Gilbert's potteroo of all things. So another little hopping marsupial in Australia. And these get transmitted by ticks. But what's extraordinary is it doesn't make as far as we know, it doesn't make the wildlife sick. And that's probably because they've been getting to know each other and co-evolving for a very, very long time.
What size was that?
So that's a red blood cell there. All these things are red blood cells.
So is that, I can't see that properly. Is that a worm, or what's the shape of that?
It's round. Well, it's pear-shaped actually. The word paraplasm is Latin or Italian for pear. It comes from that, and sometimes they look like pears. They don't always. But if you want to know the size, well, it's about 7. Well, the red cell's about 7 microns. It's bloody small. Now, so I've told you about the ticks that we have in Australia. There's 65 of them or so, different types, and they've been here a very long time. What has happened in the last 250 years or so, since European settlement of the continent, there's been a number of ticks that have been introduced. But only 5. And these are ticks of cattle and ticks of dogs. And they've come in, of course, with the cattle and the dogs that have been brought to Australia. They've only been here 250 years. They weren't here before that, because there were no cattle and no dogs in Australia prior to 250 years ago, unless we count the dingo, which has been here a bit longer. And they brought with them a whole load of cattle parasites and dog parasites. But what is important is that, to the best of our knowledge, those 5 species of tick did not bring any of the major zoonotic diseases, the major northern hemisphere infections that are so well known. These are important parasites, but they're veterinary parasites. They're not medical parasites.
I thought you said that bottom one was flu fever.
Yes, we find that. So Coxiella, indeed, is found in the brown dog tick, which came in as well.
Humans get Q fever.
And humans get Q fever. That is true. But these others here are not transmitted to humans, as best we know. Okay, so how do people get infected? I think you know that if people walk into the bush where these life cycles are all occurring, they get the tick on them, and the tick can transmit the infections which normally live within the native animal populations. Okay, so what organisms could there be? Let me introduce you to some terms. You understand the term microorganism is a word that means a very small bug. We talk nowadays in terms of microorganism communities. So that's a community of bacteria or a community of protozoa or a community of viruses. And you'll also see these two terms which are being used more and more. So the microbiota or the microbiome. And all these words mean is that micro means very small, and the biota or biome refers to the sort of population of organisms. And I'm going to talk, and Charlotte's going to talk in a moment, about the microbiome of ticks. And what we mean by that is the community or the population of organisms, bacteria, that live inside ticks. You might have heard that the microbiome of humans is becoming more and more researched and more understood in its effects on everything that we do. So we have a microbiome inside us, our gut microbiome, that researchers are now understanding affects us in many different ways. And it's probably the same with ticks. So you've seen a picture a bit like this. Here's the inside of a tick, and you can see its gut and its ovaries and its salivary glands. And within these live things called endosymbionts. These are bacteria, organisms that live inside the tick and actually make the tick healthy, would you believe, in the same way as our microbiome makes us healthy. So there's lots and lots of organisms that live as endosymbionts within ticks. But what happens is, over hundreds, thousands, millions of years, some of these develop the ability to be transmitted from the tick into the host and back again. And they then become termed the pathogens. And the theory is that all the pathogens that are nasty for us evolved originally from organisms that just lived inside ticks. So there's a number of these. They if you like, rogue microbiome organisms, and they can be very pathogenic in some hosts. And then there are bugs on the outside of the tick that are contaminants. That's just a picture with lots of funny lines on it that shows all the different sort of bacteria that you can find inside a tick. There's hundreds of different types, okay? And this work's been done overseas, but this is now what we're doing with Aussie Ticks. So that's what we'll talk about next. So we've been researching a tick-borne disease potentially of people for the last 3 or 4 years, and we've been receiving ticks from all over the country. We've had thousands and thousands of ticks. We identify them. Remember, one of the things we really wanted to make sure about was that we didn't have any of those introduced ticks, or if we did, we knew about them, those introduced ticks from the northern hemisphere that cause disease in people. We know we've got some introduced, but we haven't yet found any of the Ixodes ricinae group, that group that transmits Lyme disease, for example, here in Australia. So I'm going to hand over to Charlotte, who's going to tell you a little bit about the molecular biology tests that we're doing.
Thank you. I don't know, this isn't designed for females wearing a dress. I'll just put it here and hopefully I don't walk away too far. Hello everyone, I'm Charlotte Oskam. You might have seen me on the SBS Insight or also the ABC Catalyst. I do apologise for the Insight interview. I actually flew over and lost my voice completely. So they were feeding me up on hot lemon tea and all that sort of stuff and honey tea so that my voice would come right. And then they came to me and my voice just still wasn't there and I was coughing the whole time. And anyway, hopefully it came across okay. And I'm standing here today a week after spinal surgery. So hopefully I can keep on track. I'm taking quite a few different medications at the moment. I've got a bit of brain fog. Because I had a bit of my intervertebral disc cut out of my back. So hopefully I don't collapse or anything, but I think I should be fine. The drugs will get me standing. So I'm going to probably talk about the most tricky part of our research, thanks Peter, in my state. So I'm going to talk about some terms that you might be familiar with when you're sending samples away, and that is the PCR, the polymerase chain reaction. I'm also going to talk about what our research uses, and that's next generation sequencing, which is pretty much the next sort of technology that allows us to look a little bit deeper into your samples that you're sending to us. I'm also going to tell you a little bit about some of the findings from our research to date, particularly some of you might be aware we have found a unique Borrelia in some of the specimens, in some of our ticks. So what is PCR? Essentially it's basically like photocopying the genetic material. So some people might refer to it as molecular photocopying, but it isn't really in a sense, because it's not just taking one copy after each other. What it's doing is it's actually targeting a specific region of our DNA, of our genetic code, and what we term as amplifying, making many copies, but in an exponential sort of way. So we call this sort of the sample that we get from it is an amplicon, or a targeted sequence. So basically what polymerase chain reaction is that we start, is that the laser? So dull. Okay, so basically what we do is we start with our original template strand, which is in the red there, and we, for this example, we just have one copy. So one copy of DNA. DNA is double stranded, so we have two strands. So what we do with the PCR is that we have a number of different temperature gradients that we put the DNA through, and the first part is actually separating, or what we call denaturing, the DNA. So we separate, say, the left and the right strand, and they become our template strands for where we have nucleotides, little bits of DNA. You might have heard of A, C's, T's and G's. They attach to them and they create the second round. So we now have two copies of DNA after our first round of PCR. And so we go through a number of different cycles, and say after our third cycle we then have 8 copies of DNA, and every success of each different round of PCR, that DNA becomes a template for the next round. So what do we have in a PCR reaction? We have DNA. So my background is actually in ancient DNA. I'm a molecular biologist, and in 2012 I finished my PhD looking at ancient DNA and the moa from New Zealand. If you might have picked up, I've got a bit of a New Zealand accent, or a lot of a New Zealand, a bit of a New Zealand accent. Peter loves to sort of rib me for that. So in 2012 I met with Peter because I was using this thing called next generation sequencing, and he wanted to see how he could use it for ticks, and I've been working with him ever since because I think it's just fascinating and I want to know more, and you guys obviously want to know more as well. So we add DNA to the PCR. It could be DNA that we've got from urine or from blood or from bone or from fossil eggshell or from hair or even soil or ticks. We use tick DNA a lot. We have specific enzymes that allow the PCR reaction to take place, where it can actually add extra bits of synthetic DNA so that it can be copied. The synthetic DNA is the nucleotides that get attached, and we also have stabilizing reagents. Basically it's almost like the stock in your soup. It sort of brings it all together and makes sure that all the enzymes and the DNA and everything is sort of working together. Now we also have a thing called primers, and if you've been having your samples sent away, or if you've been taken notice of the Senate inquiry and other things like that, you might have heard the term primer. Basically what a primer is, it's a synthetic piece of DNA, anywhere from about 15 to 30 bases, so A's, T's, G's and C's in a specific order. And they're designed to target a specific region of DNA. So it could be targeting a specific chromosome that you want to amplify, or a particular gene. Now these primers are really important to pick the right area, because you want to make sure that if you amplify a targeted region, then you can actually compare the DNA that you've amplified to something that's in a database. So we need to make sure that we're targeting a specific region that's going to be informative. So we've got 8 copies after cycle 3. After 30 cycles, we have 1 billion pieces of DNA. So essentially, PCR is really sensitive. It's more sensitive than cloning. So the old days, they used to bash up a tick, and they used to do cloning of the bacteria DNA. But the thing is, a lot of the bacteria can't actually be cloned. It can't be cultured, sorry. So a lot of bacteria can't be cultured, and PCR is sensitive enough that it will actually, if there is a bacteria there, even if it's just 1 copy of DNA, after 30 cycles there'll be 1 billion pieces of DNA. So I'm used to working with really low copy DNA, because I was able to get DNA from a 20,000-year-old emu eggshell from Australia. So that's why I'm sort of working on your samples and on ticks, because I really like a challenge. If there's something there, I really want to find it. We also talk about PCR efficacy. So having a really successful PCR is really determined on how specific it is, how efficient. So are we getting lots of DNA? There's no point doing a PCR if we're going to get very low numbers of DNA at the other end, and how accurate it is. So basically we have to have a think about what we're actually doing in our PCR, what our outcome is, because as soon as we start to go into 40, 50, 60 cycles, yes, we get lots of DNA sequences, but the more times we do it, we're actually going to increase the amount of errors. And also with primers, they're heat dependent. So if you have a low temperature in your PCR, it's more likely to bind to something non-specific. So if you're trying to, say, amplify a blood meal, trying to find out what a tick has been eating, and maybe it's been eating a kangaroo, but you've also touched the tick, so there might be human DNA there as well. If the primer is really specific, hopefully it will just pick up the kangaroo DNA. But if you reduce the temperature, for example, you made it less specific, then you're going to pick up human DNA. So you start to get non-specific amplification the more PCR rounds that you do. And so even with ancient DNA, we don't go anything sort of past sort of 40 cycles, because if we've got 1 strand of DNA in our sample, we'll get 1 billion sequences. So there's no point doing lots and lots of cycles, because we just get error. One other thing is that we actually get a tapering off of reagents, because we only have a certain amount of enzymes, a certain amount of other buffers and things like that. So the more we do, the less accurate our samples are. So we try to keep within a really good window so that we get really good accurate sequences on the other end. So let's have a look at next generation sequencing. Peter's already talked about the microbiome being a community of microorganisms. I'm going to talk about the tick microbiome and how next generation sequencing can allow us to see what is found within the tick. So the difference between next gen sequencing and PCR is that high throughput. We're able, with the PCR, you just get a single sequence and from 1 sample. with next-gen sequencing, you get millions of sequences from many samples. So, and that's because we're actually trying to look at all the bacteria, for example, in the sample, as opposed to just looking for one bacterium. We're looking for the whole community of bacteria. So with NextGen, it ends up being faster and cheaper because we're able to look at lots of different things at the same time. It's more sensitive because it ends up copying the DNA quite a lot of times and is able to look at low copy number in a community as well. However, it is less specific. So instead of with PCR, we're looking for maybe just Borrelia. With next-gen sequencing, we're looking at all the bacteria. So if there's Borrelia there, if there's Ehrlichia, Bartonella, we'll be able to see the whole lot. So that's a good thing. One of the downsides of next-gen sequencing is the length, how long the piece of DNA that we're actually looking at. So we're limited by the length, and that's why when we do our research, we couple the next-gen sequencing, where we can see the whole community, with PCR, when if we do find something interesting, we can then go and design something else to look at a longer region of DNA. And I'll give you an example in a moment. So we do a thing called 16S metabarcoding. We're looking at the 16S ribosomal RNA gene. That's ubiquitous. It's found within all bacteria. So that's one good thing about our technology, is that we're using a gene that is found in all bacteria. Sometime with PCRs, you want to have a really specific PCR, and say with Borrelia, you might just want to look for the Borrelia's got a flagellate, it's got a tail, so you might want to look just for the gene of that tail, which is the flagellin gene, which I'll speak about as well. But with this, because not all bacteria are going to have a flagellum, it's not going to work for everything. So with NextGen, we're looking at one particular gene that has that's found in all bacteria. So we have this technology where this is an example similar to what we had before. We had the one copy number of DNA, and we separate it, and we're going to copy it. With next-gen sequencing, I'm not sure if it comes up, but if you see there's an ID index, there's a unique identifier that we incorporate into the PCR with each sample. So what that does is allows us to look at all the bacteria found within that tick. They all have that same unique code, and the same with this one, has a different unique code, and so on. You can replace those ticks with, say, your blood or your urine samples, for example. So what this allows us to do is pull all the samples together, put it on this fancy machine that goes ping after 36 hours or so, and it gives us millions of sequences. We roughly get about 15 million sequences from one particular run. We aim for about 120 samples at a time, so that we get a lot of coverage. We get a lot of DNA sequences per sample. And so what did that give us? Well, this isn't very interesting, isn't it? Is it? It's a big pie graph. It's pretty much one colour. It's pretty boring. We were very disappointed. We found out that when we first started looking at the paralysis tick from the northern beaches, where a lot of people were presenting with a Lyme-like illness, these ticks, they had about 95% were filled with a bacterium called Candidatus midichloriae mitochondriae. Is there any Star Wars fans out there? You're seeing the Phantom Menace, Star Wars #1?
Master, what are midichlorians?
Midichlorians are a microscopic life form that resides within all living cells.
They live inside me.
Inside your cells, yes. And we are symbionts with them.
Symbionts?
Life forms living together for...
So George Lucas really knew about things that are found in ticks, didn't he? So we've got Qui-Gon saying, to Anakin, you've got this life form in you, and there's this life form which was called midichlorians. Now, our colleague over in Sydney, Nate Lowe, he found a bacterium that was found in mitochondria. So mitochondria in your body are like the little batteries, the little powerhouses in your body that creates power. It creates energy for your body. And so he actually wrote a letter to George Lucas and asked, can I please name this bacterium that I've found in mitochondria? Can I call it Candidatus midichloriae mitochondriae? And George Lucas said yes. I've seen the letter. It's only in his drawer. He hasn't even put it into a photo frame or anything yet. I'm surprised. So anyway, so this led to our paper. And what we did is we actually blocked or inhibited the amplification of this bacterium, because surely there's more than just, one bacterium found in ticks. And so we've got this sort of graph here. Basically, it's showing that when we block this bacterium, we get an increase of the diversity of bacteria found in the ticks. And that's really great. So when we block the midichloriae, we get, 50 shades of grey, but there's a little bit more to that than this 50 shades of grey. We've still got a little bit of the midichloriae mitochondriae DNA, but that little red thing is what I'm going to talk to you about. And we found a Borrelia DNA sequences, and this Borrelia was from a paralysis tick. And when we looked into our database, we found that this paralysis tick had come from an echidna, which is really strange. We're like, how did we get just one single tick out of 196 from an echidna that just happened to have Borrelia in it? We just thought that was kind of strange, but really, really cool at the same time. So we got my PhD student, and we're going to talk about this in next. So this sort of led to the catalyst episode with Aruba, the echidna from up in. So you're just as bad as me. From up at the wildlife sanctuary. Kenyana, yeah, wildlife. Sorry, Peter, to put you on the spot. Kenyana wildlife, there's the echidnas up there, and we've got, I think it's Mark that's videoing it there. So that was on the Catalyst episode. And this was all because of Kim, my PhD student. She had a look at echidna ticks and found that about 40% of them from the East Coast had this new Borrelia. Now, I was talking to you a little bit about the limitation of next-gen sequencing is that really short fragment of DNA. Now, one thing to note is when you only have a short fragment of DNA, you've only got a short bit to compare against. And so you don't really get you can't really discriminate, you don't really get the ability to see how different, because you might be looking at a part of the DNA that's very similar. So it becomes very difficult to distinguish between certain species. So initially we thought, in Alex's paper, we thought that the Borrelia was more closely related to the relapsing fever Borrelia, which are more associated with soft ticks in Africa and parts of the Northern Hemisphere. But when Kim actually started to look a little bit deeper and look a little bit longer, what she found is that the Borrelia actually is a new group of Borrelia, in a new species in the genus of Borrelia. So just a little recap of Borrelia around the world. We know we've got this Lyme disease group, which is Borrelia afzelii, Borrelia burgdorferi sensu sensu stricto, all those different species. And I think there's about just over 20 or 21 recognized Borrelia burgdorferi sensu species now. There's the relapsing fever group, which is shown at the top, more associated with the soft ticks. There's quite a few, maybe as well as 15 different species of those. About 2003, they found a new group of Borrelia that was in ticks from tortoises. So, and they termed it the reptile group. And then our one is found in the echidna ticks, and it forms the 4th group. So genetically distinct from the Lyme disease group, sort of more closely related to the relapsing fever and the reptile group, but distinct enough genetically that it forms its own group. So at the present, we still don't know if it is pathogenic or if it causes disease. Peter mentioned before that a lot of bacteria, they evolve in the ticks as well as the wildlife and might not cause any disease to the echidna itself. But thanks to you guys, tweeting, Facebooking, this all helps with the sort of standing of our publication. Our paper was in regards to profile and sort of publicity and all that sort of stuff. It was number 1 out of 175 papers published in that journal at that time. It's number 5 out of 2,000 for all the papers published within that journal. And it is in the 97th percentile for all papers published in regards to, the publicity of papers. So having this sort of status on a paper is really good for us because it shows that we're got an impact, not just in the research society, but also in the public and around the world. So we have people tweeting and Facebooking all around the world, and news stories too. Now, a couple weeks ago or a month ago, Kim published her 2nd paper. She basically looked at this Borrelia in more detail, and yes, it's still that 4th group, but we've actually been able to name it. We've called it Candidatus Borrelia tachyglossae. Now, tachyglossae is the genus name for echidna. So the echidna's name is Tachyglossus aculeatus, and tachyglossus actually means fast tongue, if you wanted to know. So this Borrelia has been named, and we're doing a little bit more research into that as well. So this is Kim on the right. She's holding a koala at a conference last year. Her research is really looking at all those sort of endosymbionts that are found in ticks, in wildlife ticks specifically. She's looking at the Borrelia. She's already sort of done that. She's published 2 papers. She's now looking at pyroplasms, and she's starting to identify some really cool new things. So I'll be sure to update our Facebook page as papers start to come out from her, but she's coming towards the end. She's only got probably a year to go, if that, for her PhD, and she's done very well. Now, I've got an honours student at the moment. I mentioned about my background in ancient DNA. I've got access to all the ticks in museums and national collections around Australia. So I've been able to go in and, sort of borrow a lot of the ticks, and we ended up having 850 ticks from echidnas sent to us from all these museums, and she was able to find Borrelia, but only in the Bothriocrodon concolor ticks, so the specific echidna ticks has the Borrelia. Although other ticks do bite echidnas, she wasn't actually able to find Borrelia in the other ticks. So what that means, we don't know. We need to have a look a little bit more. But yeah, she was using similar methods to what Kim was doing. And we also, at the conference in Brisbane last year, I had the opportunity to dissect 7 echidnas that had been frozen in the museum for the past 20 years. That might sound gross, but yeah, it is gross anatomy. And we're able to find Borrelia for the first time in the skin of an echidna, just at the site where the tick was biting. So whether the tick was actually receiving the Borrelia from the echidna, or whether the echidna was receiving the Borrelia from the tick, we don't know. But it's definitely exciting results anyway, and that's from her honours, which was meant to be due on Monday, but because I'm sort of having difficulties with concentration, she got an extension, so it's not due to the week after. So yeah, I think that's all I've got to say. Yeah.
How are you all doing? You're very patient. We've got about 10 minutes to go. Is that all right? Thank you very much, Charlotte. You might have difficulties, or you might say you have difficulties in concentration, but I think that was fantastic. I just want to finish off with another couple of bits of research that we've done. We were very interested to know whether we could find Borrelia burgdorferi in Australia. I know there's lots of controversy about this. We looked in dogs because dogs can act as what's called a sentinel. They run around, they get ticks on them. They get ticks on them much more than we do, actually. And in places where Lyme disease exists, the dogs all have been infected. So, and in fact at a much greater rate too than humans. So if you look at the dogs, you can tell if Lyme disease is in an area. So we did this here in Australia. We had dogs. We were testing their blood, like the tests that are done in humans now. These are serological tests. These are not molecular tests. So looking for antibodies to Borrelia. And we looked in dogs owned by people who had Lyme-like illness. We looked in these foxhounds that are used to produce the anti-venom for the paralysis tick. We're very fortunate in Western Australia. We don't have paralysis ticks, but over east it's a major killer of dogs and cats in particular. It makes people sick from the paralysis as well. I won't go through this in great detail, but just to say that we did the sort of testing that is done on people when you send blood away either to Australian laboratories or to overseas. You know, we did these things. You'll be familiar with some of these terms: line blots, western blots, that type of thing. And what was interesting, we tested well over 500 dogs. In fact, you know, we tested more than that in the end. But we found we were testing for Anaplasma, Ehrlichia, Borrelia, and Diarrhea heartworm. This is a test for dogs, of course. And we hardly found any positives. Just one Anaplasma, which we think is most likely the dog Anaplasma, as opposed to the one that you might have read about or you might know about that causes disease in people, Phagocytophilum. We don't know, but there was just the one positive dog. But this testing down here relates to Borrelia, to Lyme disease. And what's really important, I think, is that we tested these 549 dogs, I think it was. 431 of them were negative. So this is a test. This is the ELISA, if you like, ELISA serology. And less than 99.9 means they're negative. But importantly, we had 118 that had a very weak positive test to Lyme disease, if you like, to Borrelia, and 4 dogs that were a bit stronger, actually, stronger positive, and then 1 dog that was very, very positive to Lyme disease. And we thought, goodness, that's a really important finding, until we found out that that dog had been vaccinated in America before it came to Australia. So that dog, that reaction, that strong positive, as you would expect if you'd had Lyme disease, if this dog had Lyme disease, it was actually because of a vaccination. But that leaves these other cases, and so there's 118 and 4, so that's 122 dogs in this. Yeah, it's 21.4, I think. Yeah, that are positive, weak positive, but nevertheless classified as positive. And yet, given the other tests that we did, especially what's called the C6, where none of those dogs were positive, we believe that the most likely explanation for these is a cross-reacting antibody. That it's not Borrelia burgdorferi, it's not Lyme disease, but these dogs are seeing, that their immune system is seeing other bacteria. Could be a Borrelia, but I think it's more likely to be other bacteria related to Borrelia. And I say this because many people in this room have probably been diagnosed or given information that they have antibodies to Borrelia burgdorferi. And our study is similar, except that our study is much clearer, I think, that we don't have, the dogs are not reacting to classical Lyme disease. So they're reacting to something. With the ELISA. Yeah. So that's something that I think tells me that there is tick-transmitted disease going on in this country, going on in the dogs, and of course going on in people too. And the question, what is it? We've heard from Charlotte about those two bugs. Just very quickly, we've also found other bugs, one of which is called Neoerlichia. Okay, we found that in 11.25% of paralysis ticks. Okay, so over east. And it's very similar to these two bugs here, Neoerlichia micurensis and Neoerlichia lotoris. And if you go and find out what they are, micurensis is a well-known pathogen in Europe in particular. It comes from ticks off hedgehogs. And this is an emerging important disease in Europe. But what is interesting also, lotoris is found in ticks off these guys. These are raccoons in America. And this one is not thought to be pathogenic. And I guess that's again something I wanted to say, that we found a Neoerlichia in ticks in Australia. Overseas, some Neoerlichias deadly, pathogenic to us, and others are not. And this is the problem we now have to face. We have to work out whether what we're finding is pathogenic to people or not. And we have now also found Anaplasma and Ehrlichia in Australian ticks. Remember, this had never been known before. So Anaplasma in the West Australian kangaroo ticks, up at Yanchep and places like that, and Ehrlichia also in other parts of Western Australia. 17.4% of the ticks on Barrow Island were positive for this Ehrlichia. Okay, so it depends a bit on, it varies about where you are in Western Australia. And what's interesting about these is, on the left here, this is one of these diagrams that the molecular biologists like Charlotte used. These are all the different bugs, and these arrows here show where they all fit next to each other in a sort of family arrangement. And so these are all the well-known ones, and these are all northern hemisphere organisms, and some of them are very pathogenic. The guys that we're finding here, our Anaplasma, our Neoerlichia and our Ehrlichia, are not the same as the ones in the northern hemisphere, but they're related. And so we're not finding the ones that cause people to be sick in Australia, but you can see in the northern hemisphere, but you can see how these are so closely related. These are probably, very possibly, a cause of illness in people. So, nearly finished. So why are these findings important? It's the first time this has ever been done in Australia, to find these organisms in the native ticks, and therefore opening the door to the possibility they cause disease in people. They're related to the ones that are known to be nasty overseas. I guess they could be the cause of the antibodies that many of you and many people who are ill have the reactions to. So what are we going to do next? There are two studies or two research questions, if you like, that we're asking. Which of the bugs that we're finding in ticks is transmitted, or which are transmitted into animals and people? You see, our tests can't tell us if they're pathogenic or not. We find them in the ticks. They might be endosymbionts. They might just be living in the ticks and causing nobody any harm at all, and being good for the tick somehow. We've got to work out which ones get out of the tick and into people. We need the DNA evidence of the bug in blood, skin, or urine of people. And then we need to say, well, okay, we can find these bugs in people, which actually makes them sick. Okay, so what's next? We're going to continue our work with the organisms in the ticks. We have to do the basic biology, the basic science. We're looking, as many of you know, we're now looking for the bugs in the blood and the urine of people with chronic illness. That study is underway now, and I have to make an apology here. We put all this information out, we put stuff on our website, but it has been confusing. And we're by Monday, by early next week, our whole website and all the forms are going to be updated, because we understand that a number of people have had some difficulty either getting doctors to sign, although that might always happen, I suppose, but also knowing what to do. So we've updated everything. That study continues, but the next thing we hope to do, and we're hoping to get this funded, is once the new tick season starts in August, is we're interested in getting samples from people who are bitten by ticks, and getting a bit of them, bit of the skin, and a blood sample as well. So that study will become more publicized fairly soon. So we have this website that you can access, and as I say, this is about to be updated. It will be next week, but we have new forms. Some of the forms are at the back, but we have updated all these, particularly the information for patients. That means you. It's much clearer now, I think, what we're expecting. And then you need to print these out, take them to see your doctor, get the doctor to sign the pathology request form. We can send all this information out to you by email, and you need to sign the consent form, and then the laboratory will do the test, and they send the stuff directly to us. They don't. It goes through Western Diagnostic Pathology. They don't do anything with samples or anything with the paperwork at all. It all comes straight to our lab. It's just that we need a medical courier system to do it. All right, you've been very patient. I think Stephen said we'd go for 45 minutes, and that's an hour and a quarter. So apologies for that. I hope it's been interesting, and I hope it helps you understand what we're trying to do. Ultimately, we are trying to find the cause of what makes people sick in Australia, and we're doing our very best. Thank you.